We continue with our second session for this morning with a presentation of Mr. Moses Kwase Aidoo. Moses is from Ghana. He finished his bachelor degree in agricultural technology in the University of Development Study. And he worked as an agricultural extension officer in the Ministry of Food and Agriculture in Ghana. Moses, please.
Thank you, Shuki, and welcome from the break. The topic of my research exercise is characterizing the effectiveness of chickpea rhizobium isolated from Israeli culture collection. This work was supervised by Professor Alicia Tel-Or here in Hebrew University, and then Professor Kapulnik, a researcher from Agriculture Research Organization, Volcanic Center. My name is Moses Kwame Aidoo. Rhizobium is a useful gram-negative soil bacteria that can colonize a legume's roots and symbiotically can fix nitrogen. Rhizobium can form nodules on legume's roots and reduce atmospheric nitrogen to ammonia, which can be utilized by its host. Rhizobium can again provide enough nitrogen that can meet the physiological needs of the leguminous plants. It is important to note that every rhizobium, every leguminous species do have an effective and a specific rhizobium group that can establish effective nodal fixing nitrogen on its roots. Inoculation is the process of introducing the isolate or the rhizobium into the leguminous plants to make sure that the plants will get enough rhizobium to colonize the root system so as to prevent re-inoculation during the later stages of the plant. Rhizobium strain used for such inoculum should be able to survive in a large quantity as a free-living soil bacteria. A better fixing nitrogen can be achieved when a superior and effective rhizobium is used for the inoculum. There's a whole lot of factors that affect the rhizobium inoculation and then the quantity of nitrogen that it's faced. And among these factors is diverse agroclimatic conditions, cropping system, and cultivation practices parameters. The most common mechanism of infection is through the root hairs. And what happens is that as soon as the rhizobium penetrates the root hairs, the point of infection stops growing, and then it begins to kill, forming nodules. This diagram summarizes the penetration of the rhizobium into the root system of the chickpea. This is a chickpea seedling, and this is the root being infested with the rhizobium. And what's happened is that as soon as it affects the root hair stop growing and then begin to curl in, forming the nodules. And then the infection thread begins to form. And this infection thread grows towards the cortex of the root. And once the thread is infested, it carries the bacteria into the cortex of the roots, ensuring that the fixing of nitrogen becomes effective. Chickpea is a leguminous plant and is scientifically known as Cicer arietinum, and it has a plant height of 20 to 50 centimeters. It has a small feathery leaves on the both sides of the stem. It has a seed pod containing 2 or 3 seeds. It also has a white flowers with blue veins. For optimum growth and yield in both tropical and subtropical, chickpea needs more than 400 millimeters of annual rainfall, and these should be well distributed. In fact, chickpea is a very, very important crop in the Mediterranean area, and then in the world, it is ranked as a third most important crop. A lot of food can be prepared from chickpea, and it contains 25.3 to 28.9% of protein. Among the food that can be prepared from chickpea are hummus, snacks, soup, sweets and condiments, and also sprouted seeds and fresh seeds can also be eaten as vegetables. When the two come together, when the chickpea, being the leguminous plant, and the rhizobium come together, symbiosis are formed. And what is happening is that it leads to the formation of root nodules that can fix nitrogen and later convert it into ammonia. And this ammonia is subsequently also converted to amino acid, which form the building block of protein. The energy that is needed to drive the energy fixing processes is carbohydrate, and this carbohydrate is produced in the shoot of the chickpea through the process of photosynthesis. And after that, the carbohydrate is translocated back into the root nodules and used as an energy to drive the nitrogen fixing enzyme nitrogenase. This enzyme, that is nitrogenase, is very sensitive, or is extremely sensitive to a high temperature of high oxygen concentrations, and it can be deactivated or stopped functioning. And this can activate again by reducing the high levels of oxygen in the root nodules. And this lowering of the high levels of oxygen in the nodules can be achieved by the substance called hemoglobin. The diagram here shows how nitrogen is fixed. This is a root nodule. It contains millions and billions of rhizobium cells that fixes nitrogen. So what it does is that it uses sugars that is produced from the shoot of the leguminous plant or the chickpea as an energy, and in return fix nitrogen and can then convert it to ammonia and then amino acid that can be used by the plant for its development. So it's like give and take. The objective of this study is to select and test for the most effective chickpea rhizobium that's available in Israeli rhizobium collection. And to help us to achieve this main objective, two specific objectives was set. The first one is to determine the contribution of each isolate on the chickpea rhizobium, and the second one is to determine the rate of which each rhizobium contributes to the formation of root nodules. This research was conducted in the laboratory and in the greenhouse at the Agricultural Research Organization, Department of Agronomy and Natural Resources, the Volcanic Center. 8 strains of rhizobium was also collected from the same institution and tested in this study. And then a chickpea, a local variety called the Havate, which was healthy, high vigor, and free from foreign materials, was also used to test the effectiveness of the isolate. East extra monitor broth and agar was used to grow the bacteria, and then in the laboratory experiments, the isolate or the bacteria was grown initially in 250 flasks containing 50 ml YEM broth media, and later incubated at 28 degrees Celsius on a rotary shaker. And later, this medium was used to inoculate other medium, and a dilution of rhizobium cells with a sterilized tap water containing 10:1 up to 10:8 microliter cells of the rhizobium were grown on the YEM agar at 28 degrees Celsius in incubator. And then later, the the isolate was taken into a greenhouse to check for its effectiveness. And in the greenhouse, the experiment was divided into two. The first one was replicated at 10 times, and we sampled twice. We sampled twice to check for the chickpea contribution to the growth of the chickpea. And then experiment 2, it was replicated 20 times, and it was sampled at 4 times to also check for nodal formation. Randomized complete block design was the experimental design used for this experiment, and then the experiment was laid on a wire mesh benches to prevent contamination, cross-contamination of the isolate. And then the pot was also well spaced out to prevent the touching of the plant for the same reason. And you can see that the base of the plant is covered with a sterile perlite to also prevent the stubborn isolate from jumping from one pot into the other, if it's possible. 4 seeds were sown in each 10-liter pot, and 1 ml suspension of rhizobium isolate containing 10 raised to the power 7 cells was inoculated to each seed. And then later, 2 healthy plants per pot were retained. And were retained after the formation of the first trifolia leaf. Plant height, number of nodules, nodal dry weight, shoot dry weight, root dry rate, single dry weight of nodules were the parameters that was measured in these studies. And a statistical software, JAMP9, was used to analyze the data. In the laboratory, we check for the growth of the isolate. You can see that isolate 2, 5, and then 8 have a similar trend, especially in 42 and 68 hours. But isolate 5 continued to grow at 140 hours, and isolate 2 and 8, the growth reduced at 140 hours. Isolate 1 and isolate 7 also have the same trend. At 42 hours, the growth was very slow, and then the growth picked up at 68 hours, and then declined at 140 hours. Of course, you can see that the decline of this is higher than the decline of isolate 1. And isolate 3 growth at 42 hours was very slow. It picked up in 68 hours and continued to grow at 140 hours. And isolate 4 was very slow in growing at 42 hours. Growth picked up at 68 hours, and it sharply dropped at 140 hours. And isolate 6, the growth of 42 and then 120 hours is similar, but the growth of 68 is very low, and this was attributed to the fact that it may be due to a sampling error or some condition which was not favoring the growth of the isolate. I must say that I must apologize for not having the error bars. This is the kind of an experiment that only one sampling was done in these hours. In the greenhouse, this picture was taken in the greenhouse, and it shows a chickpea plant that is inoculated with the isolate, and the chickpea plant that is not inoculated with the isolate. And here we can see that the root is very dense, full of nodules. The shoot is big, and then the stem is long, relative to the uninoculated one, which is A, which have small roots, and you can hardly see a root nodule here, and it have a short stem, and then the shoot is also small compared to the B, which is the inoculated one. In the greenhouse, for the first experiment, the sampling was done twice, like I said, and then the first one was done at the vegetative stage, And the isolate's contribution to the parameters checked was so amazing. At plant height, it shows significant, all the control shows significant difference all the isolate shows a significant difference to the control. And then statistically, there's no difference among the isolates. But isolate source contributes to the high growth of the plants or high plant height, followed by isolate 3. And you can also see that all the isolates contributed more than 30 to the plant height, except isolate 1. And then shoot dry weight, it also shows significance among the isolates and then control. Isolate source again was the highest, followed by the isolate 7, and then isolate 8. And then the root dry weight, it also shows significance among the control and then the isolates. Isolate source continue to lead, and then isolate 8, which is also similar statistically to the first one, which is isolate source. And then number of nodules show a high significance among the isolates and then the control. Statistically, as you can see, among the isolates, they are all the same, but isolate 1 contributed the highest, followed by isolate 5, and then isolate 4. But like I said, they are all similar statistically. And then nodal dry weight, it also shows a high significance among the isolates and then the control. And then isolate 1 and then isolate 7 was the leading contributor to the nodal dry weight. But again, there's no significant difference among the isolates. And then single nodal dry weight also isolates source gave a higher weight, followed by the nodal. One thing we observed that though the number of nodules developed by the control was few, but we observed that it was big. That's why it's have this kind of weight, especially when you compare it to isolate 5 and isolate 3. The plant height continued to grow at 80% flowering stage. And there again, it shows a high significance among the isolate and then the control. And then the isolate source continued to lead, followed by isolate 5, and then isolate 3. There again, there's no significant difference between the isolates. And then shoot also shows significance within the isolate and then the control. Isolate source gave the highest contribution to the shoots, followed by isolate 8, and then isolate 3. And then root dry weight at 80% flowering also shows significant difference within the isolate and then the control. Isolate source gave 80.2, which is different from say isolate 7 and isolate 3, but similar to isolate 8 statistically. And then number of nodules also continues to increase, and it shows highly significance within the isolates and then control. Isolate 8 gives the highest, followed by isolate 5, and the third highest was isolate 1. And of course, statistically, they are the same, they are no difference. And then nodal dry weights also show a highly significance to the control. Isolate 6 gave the highest, contributed to the highest value to the nodal dry weight, followed by isolate 8, and then isolate 3. And then nodal dry weight, like I said, the nodules was so big, but it was few. And then the control and then isolate source has the higher weight in the single nodule. And in the second greenhouse experiment, where formation of nodules was tested, we observed that the development of nodules have the same trends. So we sampled, we repeated it at 20, like I earlier said, and we sampled it at 4 times in these times, 4 weeks after planting, 6 weeks after planting, 8 weeks after planting, and 10 weeks after planting. And you can observe that isolate 1, 4, 5, 7, and 8 have a same trend of nodal formation. It increases from 4 weeks after planting and then hits its peak at 8 weeks after planting and begins to decline at 10 weeks after planting. But isolate 2, 3, and the 6 continue to increase in the nodal formation at the later at 10 weeks after planting. That's the later development stages of the chickpea. And you can also observe that in isolate 3, at 6 weeks after planting, the nodal formation was reduced, and this was attributed to the high temperatures in the greenhouse at the time of conducting these experiments. And then you can also observe that in the control, as at 4 weeks after planting, there wasn't any nodal formed, but all the isolates, there's a nodal form as at 4 weeks after planting, though we recorded a lower amount at 2 and then 6 and then 7 isolates. In summary, the isolates was classified into slow and fast growing, and then the fast growing isolates had a similar trend like I've already shown, and then it's multiplied and formed more single colonies with abundant extracellular polysaccharide on the YEM agar 5 to 7 days. And then the fast-growing isolate formed nodules during the early part of the chickpea plant development. All the parameters used to assess the growth and then development and then microbial colonization has a higher value. The plant height, of course, show a significant increase by the contribution of the isolate, and then the dry weight of the shoot was also influenced by the activities of the isolate. And then the lowest dry weight of the shoot was recorded by the non-inoculated control chickpea, as I showed you. And then the dry weight of the chickpea roots inoculated with the rhizobium isolate was significantly different from the non-inoculated chickpea. All the isolate produce higher nodal dry weight compared to the non-inoculated chickpea. The chickpea inoculated with isolate significantly contributed to the nodal formation and development. The nodal formation followed the same trend and begins to decline at the later stages of the plants. And the results, these results are the indication that inoculated chickpea with some isolate continue to contribute to the formation of nodules, whilst others do not. In conclusion, isolate 1, 3, 4, and 8, isolate 1, 3, 4, 8, and 6 were classified as the most effective isolates, and isolate 2, 5, and 7 were classified as the less effective isolate. And then the difference between the two was estimated to be the 30%. And then we recommend in future that the most effective isolate should be investigated more in the first situation to evaluate its effectiveness in the hardship of the environment. Thank you.
Thank you, Moses. One question that I was not sure about. Maybe I didn't understand. In the previous slides, you have presented the isolate 6 as having, as inducing enhanced growth and more nodules, etc. However, here, in the number of nodules, you show as if it is like the control.
Yeah, it's true. You are perfectly correct. That's why, if you can observe the conclusion, look at the way I put the conclusion. I said isolate 1, 3, 4, 8, and source, which means there's a question mark there. Because in the normal circumstances, it should have been 1, 3, 4, and source. But chickpea is a cold season plant, and we have only one season to carry out this experiment. And of course, you know, I have been fighting with you and Nina with time. And then there's no way we can leave this. We can wait for next year. By then, the whole exercise would have passed. Though still, we have to do it. So we decided to go to the greenhouse with the refrigerator, but with the air conditioning. But we observed that there wasn't enough sun light. There wasn't enough light to help the plants for us to get the benefit of it. So we were forced to do it under the hardship condition. The temperatures was very, very high. We tried putting fans, including standing fans, to reduce the temperature, but we couldn't successful. But in order to help us to show the trends of its development, we still went ahead to
also, this slide is from a second experiment?
This is from the second experiment where the formation of the isolate was estimated.
All right. Well, first of all, it was a very nice lecture, and I think the project went very well. And I still assume, based on the two numeric tables that you had, that number 6 made the best results that you had. So I would actually, in that respect, take the results about the number of nodules that you have here with less significance than the actual tables. If you come back to the table, okay, and look at number 6 here, no doubt that wherever you have the milligram per nodule, okay, the last row, and wherever you have the weight of the shoot and the weight of the root, I mean, the results are very clear that that number 6 does it. And take the previous table where you had even higher. Do you have the previous table?
Yeah.
Yeah, and you have a 7 milligram per nodule, and you have an enormous evident increase in shoot and in root for number 6. So I think that these results are more important, aren't they?
And this I must say that this was carried out at the right season of the plant. Yeah.
Now let me ask you something even a little bit more sophisticated. Okay. The group in Bedagan, not under Kapulnik, but the previous boss, who was one of the founders of nitrogen fixation inoculation experiments in Israel, did actually run chickpea inoculation in a way that the seeds have been inoculated with the bacteria before their introduction into the soil. What would you say about such an approach for chickpea?
Yeah, such an approach will be very laudable, I must say, because like we can see, the chickpea really contributed to the development and then the growth of the isolate contributed to development and the growth of the chickpea, which of course is a clear indication that we are going to get a good harvest from the plants. So one of the things we proposed that we even wanted to carry out, because of time and then the weather didn't allow us, is to even coat the bacteria on the chickpea and then later evaluate how we are going to get to evaluate these growth parameters.
And last question: when you go back home, what would be the significance of whatever you have studied and summarized here for any future developments back in Ghana?
I must say this is very practical. It's very practical, and anybody can really get his hands on in putting it into practice. So when I go back home, I just have to affiliate myself with a research institution or agriculture organization and see how this can be practiced.
We see some differences, for example, in the plant height.
Yeah.
For example, in the 5 and the 6. However, one plant it's higher. The 6 is higher than the 5. In the number of nodules, it is less. So, do you mean that the number of nodules doesn't like affect that much the growth as just the rhizobium development, or what it most affects the development of the plant?
The number of nodules really affects the development of the plants, because the more the nodules, it's assumed that the more the nitrogen fixation. But I also have to point out that we can not just count the nodules and also say that it's active. The nodules is positive, or is active. It is acting, or is sometimes it can be there and it can be inactive. Yeah. So if it's inactive, then its contribution to the growth of the development is also questionable.
I have a question related to the soil. What would be the differences, like the main differences between the soils for the fixation and the symbiosis to occur between the rhizobium and the legumes? Does it work the same for all the type of soils, for example?
No, it doesn't. It doesn't work the same in all type of the soil. I show that we have a lot of factors that affect these, and then you can even see that at my second experiment, the hardship of the weather, the abiotic factors, really affect the contribution of the isolate into the growth of the plants. So soils with different factors, different soil nutrients, also have different influences on the contribution of the isolates. And I must also say that even the type of the variety of the chickpea that you use also contributes into the fixation of the nitrogen by this isolate. Supposing I use seed which is less vigor, definitely it's going to affect even germination, and then it cannot grow to facilitate the nitrogen fixation of it.
So do you expect that the cultivar and the type of soil will interact with the different rhizobium isolates?
Definitely.
Namely that for each type of soil you have to test and find the optimum isolate to be used for it?
Yes.
I have one question.
All right.
What was the variety of chickpea you used?
I use a local variety called Zihavit. It is commercially adopted.
Is that commercially grown in Israel?
Yeah.
The most dominant one?
Yeah. It's
and
good. It's very good.
what about other hybrid varieties or other varieties used?
Other varieties, if it is accepted as a variety which is good, then it can also be used. Because like I said, it's the type of cultivar have a major role in the fixing of the nitrogen. So if a particular variety is certified by the seed company, that is good.
Okay, now my question is, if you had used some other varieties, then your result would have been same?
It depends. If that's have a higher vigor than what I used, the results is not going to be the same. I will anticipate a higher result than this, because it shows that chickpea heights have a range of 20 to 50, but none of my results hit 50. So which means if I use a seed which have a higher vigor, then I'm likely to hit 50 or probably a seed.
So for each variety, the isolates, the number of isolates, it might be different. For variety you use, it's like 1, 3, 4, 8, and 6. If you use other varieties, then other isolates might be effective, yeah?
It's not effective. It is here. It is you looking at the contribution. That's why I have the control, right? And look at control. Here, there's no isolate. There's nothing. Yeah. It's about how it will contribute into its development. That's what we are looking at.
Okay, thank you.
I have a small questions about the numbers, means isolate 6, and there, when there is a low number of less number of nodules, you found root dry weight less and the shoot dry weight higher. Is there any relationship between the effect of rhizobia in the shoot and root formation?
Effect of rhizobium and
the shoot dry weight and root dry weight. It means.
Yeah. There is because you know, one thing happened. We observed one thing. When it forms more nodules, after picking the nodules from the roots, then the roots becomes lighter, right? Because we have to again remember that it's the root hairs that forms the nodules, right? So when it forms a lot of nodules and we take it out, the roots become lighter. And we also have to bear in mind that the plants have to feed the rhizobium and also have to feed the root with the same starch, right? So it's like doing two things at the same time. It's not a drawback. It's a beneficial thing. But I'm telling you that it's the same root that curl, the root cells that curl to form the root nodules. So supposing the plant is actively forming root nodules, and then I harvest or I sampled, I'm likely to get a.
Okay, thank you.
I want to ask a question. Is there any possibility of utilizing such bacterial inoculation in cereal crops also, or only in leguminous crops? And what makes it difference to adapt in cereal to make symbiosis in legume and not in cereal crops?
All right. This bacteria is noted of fixing colonizing the roots of leguminous plants, and then even in the leguminous plants, we have every leguminous plant have its specific bacteria that can colonize the roots of the But what we can do to help in the cereal production is that after planting days, in the literature, it is stated that after rhizobium has been used on the field, and after harvesting, it's sort of leave the nitrogen. It improves the soil nutrition. So any plant, subsequent plants that you grow there, is likely to pick up these nutrients. So after we've cut out or we harvest our leguminous plants or the chickpea, we can grow cereals, which of course is going to help increase the yield.
Okay.
When rhizobium infects the root, does it interfere with the growth of the root hairs or not?
It interferes with the growth of the root cells.
In what way? It stops.
What happens is that when it's affected, it stops growing. The point of infection stops growing, and then as soon as it stops growing, it starts to curl to form the nodule. So it will curl, and then the bacteria will be inside, and then the root thread will develop, and this root thread will pull the bacteria into the root cortex, and this ensures a very positive symbiosis. That is, it really enhances the nitrogen fixation.
Thank you.
Last question, Hapte, please.
I haven't seen the media you used to in the greenhouse. Maybe in most cases, sterile media is used to evaluate such type of experiment. Potting soil or some kind of potting material used for your experiment, and also their effectiveness also depends on the carbon to nitrogen ratio of the media. Maybe you have to consider that one also, but in most
all right, the media used was sun. It was sun. As the sun was, it was sterilized. It was dried. As solar sterilization. That's the media that is used in the growth of the year. And then to avoid cross contamination, a perlite was just used to cover the pot to prevent the bacteria which are likely to jump from doing that.
Okay, thank you very much, Moses.